Let's run multiple sequence alignments using the Clastel-Omega algorithm, which is also known as Clastel-O. To enter the program, use the following link. We will compare the CFOS protein as found in different organisms. The program can accept sequences in different formats as input. We'll use the FASTA format, which is the most common one. In this format, the first line is a description line that should start with a greater than symbol, followed by any description, while the sequence itself appears from the second line onwards. Enter all sequences to the input text box. Please note that it is important to give informative names at the beginning of the description line, otherwise we could not differentiate sequences at the output. The simplest way to do it is by adding the G name or the organism name after the greater than symbol in each description line. Choose the protein option under Step 1. Choose the Clastel W with characters counts option under Step 2 for presenting Clastel alignment format with residue number. Click on Submit. At the output page, you will find several output files created by the program. They will be organized in different tabs. All running files and the percent identity matrix appear under the Result Summary tab. Click on the percent identity matrix link. Clastel-O first calculates the percent of identity for each pair of two sequences and presents it in this matrix. The titles of the columns are the same as in the rows, but just transposed. The most conserved pair of sequences in this matrix are the mouse and the hamster ones, with about 96% identity. The pig and the human sequences have also a high identity. The multiple sequence alignment appears under the Alignments tab. Since most sequences are longer than the 60 characters limit that can be presented in a line, the alignment is spread over to several blocks of 60 characters each. The sequences of each organism will appear at the same line in each block. For example, the chicken sequence appears in the first block as the first sequence in the alignment. Its last amino acid in this block is amino acid 60, whereas amino acid 61 appears in the next block on the left side of the first line. Please note that the last amino acid in this line is 119 and not 120 as expected because there is a gap in the middle of it. The number of the last amino acids in each organism appears at the right side of the last block, representing the length of each original sequence. We evaluate the multiple sequence alignment by looking at the conservation of each column in the alignment. There are consensus signs under each column. An asterisk is used for columns in which all amino acids are identical. A colon, two dots, is used for highly conserved columns showing only very similar amino acids. The similarity between two amino acids is defined by a similarity matrix like BLOSUM 62. A period, one dot, is used for partially conserved columns that can include also less similar amino acids. No symbol will appear under a column if it is not conserved at all. An area enriched with asterisks and colons represents a high conserved common motif, whereas areas with no symbols or areas enriched with gaps represent non-conserved regions. Click on Show Colors to present alignment with the help of distinct colors for each group of amino acids. The meaning of the standard color scheme is as follows. For example, acidic amino acids will be colored in blue, whereas basic ones will be colored in magenta. In order to remove the colors, click on the Hide Colors. Now we will learn how to create a logo from the multiple sequence alignment output. We will use WebLogo, a web-based application designed to generate sequence logos. Sequence logos are graphical representations of the bottom line of an amino acid or nucleic acid multiple sequence alignment. Each logo consists of a stack of symbols, one stack for each position in the sequence. The overall height of the stack indicates the sequence conservation at that position, while the height of symbols within the stack indicates the relative frequency of each amino or nucleic acid at that position. In general, a sequence logo provides a richer and more concise description of, for example, a binding site than would a consensus sequence. There are three multiple sequence alignment input formats that can be used in WebLogo: FASTA, CLUSTAW, the default output format of CLUSTAL-O, and FLAT. All sequences must have the same length. No digits are permitted at the input. Therefore, we need to run CLUSTAL-O again, this time changing the output format parameter under step 2 to CLUSTAW, without the number in output we requested before. Enter to the WebLogo page using the following link. Pass the CLUSTAL-O multiple sequence alignment output. Do not forget to include also the consensus signs under all columns. V-check the following parameters in order to get a clear output. Multi-line logo symbols per line 32. Enter 124 and 207 at the logo range parameter. Click on Create Logo. Now you have generated a nice logo which can be presented as a conserved motif figure in an article. Do note that the position in the logo in which no letter is displayed means that there is no conservation in that position. This could happen due to gaps or variety of amino acids at that position. You should note that the CLUSTAL family of algorithms has many installations on many sites, and user interface may not be identical in all of them. Furthermore, the interface may change and get updated from time to time. Hopefully today's hands-on experience will help you navigate this very useful program in the future as well.